As we are pursuing agriculture, we are mostly dealing with plants and its management. Okay, so plants need different type of resources for its growth and development. Mineral nutrients are one of them. Plants need mineral nutrients along with water and other resources. So how these mineral nutrients come into plant body and plant system, how it is assimilating inside the plant system, and how it is useful for plant growth and development, all these things we will discuss in this lecture. So welcome back to our channel. I am Dr. Jagdish Jena, who will be teaching, who will be guiding about this on this lecture, and we have already uploaded several lectures in our channel, so stay tuned. And if you have not subscribed yet, then you just subscribe the channel so that you will get some updated lectures. So let's dive into today's topic, that is mineral nutrition in plants. What is mineral nutrition? मिनरल न्यूट्रिशन का मतलब क्या है? What is mineral nutrition? So mineral nutrition is basically nutrient का uptake, उसका translocation inside the plant system, and how it is assimilated inside the plant system. All this total process is known as mineral nutrition. इसका मतलब क्या है? प्लांट के शरीर में न्यूट्रिएंट ऑब्जर्व कैसे होता है, प्लांट के शरीर में ये ट्रांसलोकेट कैसे प्लांट के रूट से लीफ और फॉलियस तक पहुंचता है मिनरल न्यूट्रिएंट, एंड कैसे ये मिनरल न्यूट्रिएंट जो शरीर में डिफरेंट ऑर्गन्स में पहुंचता है और कैसे यूज होता है प्लांट सिस्टम में ड्यूरिंग द ग्रोथ एंड डेवलपमेंट. दैट इज बेसिकली व्हाट मिनरल न्यूट्रिशन इज. Mineral nutrients generally available to plant in soil solution, and their uptake is mostly in ionic form. Most of the mineral nutrition, their uptake is being taken by in ionic form. One is ionic form, one is mineral form. Suppose I am telling H2O. This is a compound or complex form or molecular form. But if I say H plus, OH minus, these are ionic form. Okay, suppose one mineral, that is K plus, that is cationic form. Then NO3 minus, that is anionic form. Okay, suppose H3BO3. Okay, this is in mineral form or in non-ionic form. Okay, so some of the nutrients like carbon dioxide, here carbon, plant get carbon and oxygen from this molecule, that is carbon dioxide. H2O, here plant gets hydrogen and oxygen from this molecule. So these are non-ionic mineral, these are not mineral nutrients, these are non-ionic nutrients. Okay, and ionic form K plus, NO3 minus. Okay, these are ionic form, and mostly they are present in soil solution so that a plant can able to get them. Okay, mineral nutrients either uptake by roots or through mycorrhiza association or by foliage. Okay, so whatever mineral nutrient, जो भी mineral nutrient soil में है, that is uptaken by root by different uptake mechanism like mass flow, root interception, and the root interception, mass flow, and diffusion. So these are basically three types of uptake of mineral nutrients from the soil or soil solution, as we have already mentioned. Another one is through mycorrhiza association. That means some of the plants, they have fungal association. So what happens, if the fungus attached with the mycorrhiza attached with the root system, it actually increases the surface area. Fungal hyphae, they spread out throughout the soil system, and they extend the root activity so that they can reach to the unexplored part, and from there also they can accumulate or absorb the nutrients. So that is mycorrhizal association. Or by foliage, when we spray different micronutrient on the canopy to fulfill the deficiency symptom, or any recommendation is there to spray a nutrient solution, then the plant uptake that nutrient by foliage. Okay, so mostly artificial application, when we do the foliar application, foliar nutrition, then the uptake is taken up by the foliage. Mineral nutrition, as I was discussing, it happens through mass flow, diffusion. The absorption part, it happens to mass flow, diffusion, and root interception. What is mass flow? Again, transpiration is a process which is going through most of the time in a day. So due to transpiration, there is a transpiration pool gradient or water deficit gradient. That means atmosphere is having less water, so it pulls water from the foliage pulls water from the xylem pulls water from the root system pulls water from the soil. So there is a transpiration pool or transpiration gradient. So due to this transpiration gradient, soil water actually moves through the plant system, okay, and it lost from the canopy. So during this water flow, the soluble ions, soluble cation or anion, they moves through the plant system. That is mass flow. Then diffusion means there is different gradient of nutrients. Suppose in the root system there is low gradient or low concentration of nutrient, and from anywhere else or nearby there is high concentration of nutrient. Then from higher to low nutrient concentration there will be a movement, that is called diffusion. That cation or anion that moves or diffuse. Then root interception, where root itself extends to the nutrient, and it due to contact of root with the nutrient, if there is exchange happens between nutrient and the root, then that is called root interception. Absorbed mineral then translocated through the plant system by translocation. Once absorption happen, that nutrient translocated through xylem vessel, okay. Absorbed nutrient may take part in biomolecular synthesis, enzyme cofactor, or osmotic regulation. So that absorbed nutrient, it may remain as such, which like potassium, that do not combine or that doesn't combine with any like organic molecule, and it mostly take part in the ionic balance in the plant system. One is enzyme cofactor, that cation or anion that may be act as enzyme cofactor of enzyme. That means for activation of enzyme there is one mineral nutrient is needed. So for that some of the nutrient they are being utilized. Then biomolecular synthesis, the different biomolecules are synthesized inside the plant system. So for that also mineral nutrients are essential. So this is how mineral nutrients they are being used or assimilated in the plant system. Then one question, you might have one question that इतने सारे mineral nutrients है, there are like 118 elements in the earth, whether all those are essential for plant growth. जो भी mineral nutrients है soil में, suppose 118 mineral nutrients are there or nutrients are there or elements are there in the earth, whether all the 118 elements are essential for plant, the answer is no. Out of 118 mineral nutrients, most of the nutrients are taken up by plant or most of the nutrients are used by plant, but some are categorized as essential nutrient. Remember this term, essential nutrient. So which nutrients we are categorizing, or the scientific community is categorizing as essential nutrient? Only 18 essential nutrients are there so far, okay, and latest added is cobalt. So how they are being categorized as essential nutrients? They have to fulfill 3 criteria of essentiality proposed by Arnon and Stout in 1939 and revised by Arnon in 1945. So Arnon and Stout proposed criteria of essentiality. There were 3 criteria of essentiality. If any particular nutrient it is fulfilling the 3 criteria of essentiality, then that will be categorized under essential nutrients, okay. So that is one designation type of thing. So if you are fulfilling this thing, if you are successfully completing your master's degree or master's research and PhD research, then you will be promoted to master's or doctorate. Like that, if any nutrient that is fulfilling the criteria proposed by Arnon and Stout, then that nutrient is promoted to essential nutrient. So what are those criteria? First criteria is about life cycle. If that particular nutrient is deficient, plant cannot complete its vegetative as well as reproductive life cycle. If that particular nutrient, remember, if that particular nutrient is absent or deficient, एक न्यूट्रिएंट है जो कि एब्सेंट है और डेफिशिएंट है, अगर ऐसा हुआ then plant कंप्लीट नहीं कर पाएगा इसका वेजिटेटिव और रिप्रोडक्टिव लाइफ साइकिल. सो प्लांट बेसिकली कैनॉट कंप्लीट द लाइफ साइकिल. सो दैट इज द फर्स्ट क्राइटेरिया. Then specific role, that particular nutrient that is deficient, that nutrient deficiency can be fulfilled by applying that particular nutrient. No other nutrient is, if you are applying, then it cannot substitute the deficiency of that particular nutrient or complement the deficiency of that particular nutrient. Suppose एक nutrient deficiency हो रहा है, अगर हम उसी particular nutrient को apply करेंगे, then only that deficiency symptom will be over or that particular deficiency criteria will be fulfilled. बट एनी अदर अगर न्यूट्रिएंट अप्लाई कर रहे हैं एंड उसका क्राइटेरिया फुलफिल हो रहा है, देन दैट विल नॉट बी कंसिडर्ड एज एसेंशियल न्यूट्रिएंट. देन मेटाबॉलिज्म, थर्ड पॉइंट इज मेटाबॉलिज्म. दैट पर्टिकुलर न्यूट्रिएंट हैज डायरेक्ट इन्वॉल्वमेंट इन द मेटाबॉलिज्म ऑफ द प्लांट सिस्टम. So plant is having different metabolism like anabolism, catabolism, we'll talk later on. So that particular nutrient we are assigning or telling essential nutrient that has direct involvement in the metabolism of the plant system. So these are basically three criteria proposed by Anand and Stout, and that particular nutrient which is fulfilling these three criteria, that is called essential nutrients. So basically criteria 1 is universal. It must complete the it must be able to complete the life cycle if the essential nutrient is sufficient. If the essential nutrient is deficient, then it must not complete the life cycle. And 2 and 3 have some limitation, we'll talk later on. Nicholas in 1961 coined another term that is called functional nutrients. So basically these are the nutrients which are not essential, but along with that, actually that is not essential, but their application or their presence will help certain plants to achieve their goal or to achieve their optimum production. Okay, so that is basically Nicholas proposed functional nutrient, and some functional nutrient example is sodium for sugar beet, silicon for rice, cobalt for biological nitrogen fixation. So they are specific role, they are having specific role, but they are not essential nutrient. Okay, how to group or classify essential nutrient? We are classifying different nutrients or different suppose we are studying about seed, we are classifying seeds. We are studying about different plants, and we are again classifying plants. So basically we are doing classification to know in detail about, or to know in detail about different thing, or to study certain thing in detail. We are classifying the things. Then how we can group or classify the essential nutrients? So basically Loomis and Saul, W.E. Loomis and C.A. Saul in 1937 classified essential nutrients based on their concentration in plant tissue as macro and micronutrient. While reading the textbook, you might have heard this term micronutrient and macronutrient. So basically this classification was proposed by Loomis and Saul in 1937. So according to them, अगर plant system में there is more than 1000 ppm of nutrient present or required, then that is called macronutrient. Macronutrient are again classified into primary macronutrient and secondary macronutrient. Primary macronutrient are N, P, K, nitrogen, phosphorus and potassium. Secondary macronutrient are calcium, magnesium and sulfur. And then micronutrient, if the concentration in plant system or requirement in plant system is less than 1000 ppm, then in the dry matter, then that is called micronutrient, like iron, manganese, boron, zinc, cobalt, copper, molybdenum, chlorine, nickel and cobalt. Okay, and cobalt. Again, another classification was proposed by Mengel and Kirkby in 1978 based on their biochemical behavior and physiological function into 4 groups. So based on their biochemical behavior, how they are behaving biochemically, and based on their physiological function, Mengel and Kirkby, they classified the mineral nutrients into 4 groups. First group is the carbon, hydrogen, oxygen, nitrogen and sulfur. They are basically the major constituent of organic material involved in enzymatic process and oxidation reduction reaction. They are basically in the first group: carbon, hydrogen, oxygen, nitrogen and sulfur. Mostly they are take part in the biomolecular constituent. Group 2 is phosphorus and boron. The major role is energy transfer and esterification. Group 3 consist of potassium, calcium, magnesium, manganese, chlorine, nickel. They are mostly used in osmotic and ionic balance in the plant system. Specifically, they are associated with certain enzymes also. Group 4, iron, copper, zinc, molybdenum. Basically, they are act as structural chelate and metalloprotein, and they carry the electron from different system to system or enzyme to enzyme. Then these are 4 groups classified by Menzel and Kirkby. Then based on the mobility of mineral nutrients in the plant system, there are 4 types of nutrients. One class is highly mobile. Some nutrients are highly mobile in the plant system, like nitrogen, phosphorus, potassium, magnesium, molybdenum. So they are basically highly mobile nutrient. Okay, their mobility is highest. Then moderately mobile nutrient, that is zinc. Less mobile nutrient, example iron, manganese, copper, sulfur, chlorine. Immobile nutrients, calcium and boron. Based on mobility of nutrient in the soil, there are again 3 types of nutrients. One is highly mobile, like nitrate, nitrogen, sulfur, boron, chlorine and manganese. Less mobile, ammonium, ammoniacal nitrogen, potassium, calcium, magnesium, copper. Immobile nutrient, phosphorus and zinc. They are basically mobility in soil. So if the mobility is high, then plant can get that nutrient from a distant place. Suppose this is the plant root, and suppose one nutrient okay so this is the plant root and one nutrient present here, and it is mobile nutrient, and it can move to the plant root system, right? If it is along with water or anything else. If it is immobile like here, then the root absorption, soil mineral absorption, that will be confined to the only root surface area. So here the system is called root system sorption zone, where the nutrients are mobile, root system sorption zone. But in case of immobile nutrient, that is called root surface sorption zone, only from surface that can absorb the nutrients. Are essential nutrients requirements similar for all the nutrients? Whether जितने भी हम mineral nutrients अभी देखे, like 18 essential nutrients are there. So क्या जितने भी essential nutrients है उनका requirement is similar for plant system, equal for plant system? The answer is no. So basically we'll discuss here what are the essential nutrients, what is their form of uptake, then who discovered the essentiality and their concentration in the plant system. So you can get an overall idea कि कितना nutrient चाहिए plant को, the optimum nutrient concentration. So carbon is basically uptaken in the form of carbon dioxide from the atmosphere. It was essentiality of carbon was proposed by J. Sack, and its concentration is highest in the plant system, that is 45%. Okay. Then hydrogen, that is taken up in the form of water, H2O, and from hydrolysis of water in jet scheme you will be studied in the next lecture. So from jet scheme, due to hydrolysis, H2O breaks into H plus and OH minus, so that H is used in the plant, and its essentiality also proposed by J. Sack, and its concentration in the plant system is 6%. Then oxygen, it is taken up as water molecule or oxygen directly from the atmosphere. The essentiality proposed by Theodore D. Saussure, and the concentration in plant system is 45%. Nitrogen, it is taken up from the soil by nitrate form or ammoniacal form, and its essentiality was proposed by G.K. Rutherford, and its concentration in the plant system is 1 to 5%. Phosphorus, taken up by plant in the form of H2PO4 minus and HPO4 2 minus. Its essentiality was proposed by Pasternak, and its concentration in plant system is 0.1 to 0.5%. Potassium, taken up as K plus, and essentiality was proposed by F.J. Skimper. Its concentration in the plant system is 0.5 to 0.8%. Calcium, taken up as Ca2 plus, essentiality was proposed by Salm Horstman, and concentration in the plant system 0.2 to 1%. So like this, there are several, all the essential nutrients are here. The form of uptake, you must see all this, you must remember all this form of uptake because they are essential for your examination point of view. Then essentiality, who proposed the essentiality, then concentration in the plant system. Okay, so this is the table, you will get an idea. अच्छा, लाइक न्यूट्रिएंट्स डेफिशिएंसी डिजीजेस इन ह्यूमन को जब भी प्रोटीन, कार्बोहाइड्रेट या फैट का डेफिशिएंसी होता है या फिर वाइटामिन का डेफिशिएंसी होता है, कुछ डिजीज होता है ह्यूमन में, राइट. सो जैसे कि ह्यूमन में डिफरेंट न्यूट्रिएंट डेफिशिएंसी होने से डिफरेंट डिजीजेस होता है, लाइकवाइज क्या प्लांट में भी ऐसे ही डिजीजेस होता है? दिस इज वन क्वेश्चन. ओके, ह्यूमन में तो डिजीज होता है, लेकिन प्लांट में क्या वैसे ही कुछ डिजीज सिम्टम दिखाई देता है अगर न्यूट्रिएंट डेफिशिएंसी होता है तो? लेट्स सी. सो बेसिकली देयर आर 4 different stages of tissue nutrient concentration. Suppose this is the plant yield or plant growth, and this is the nutrient concentration in the tissue. So at the initial development stage, suppose we have shown the seed here, and up to certain vegetative stage there will be a decrease because rapid growth will be happen at the initial stage. So there will be decrease in tissue concentration in the very initial stage, and that decrease in tissue concentration is known as Steinberg effect. Okay, then there is a deficient zone where the nutrient concentration growth is higher and nutrient concentration is lower, but there is deficiency. Okay, so that is deficiency zone. And so in deficiency zone there is a part when there is nutrient deficiency but plant is not showing that deficiency, that is called hidden hunger. In the deficiency zone, that is called hidden hunger. So what is hidden hunger? When there is deficiency of nutrient in the plant but plant is not showing any deficiency symptom, then that is called hidden hunger. Then there is a transition zone and the critical range stands there. Then there is sufficient zone of plant nutrient concentration. Then there is excessive or toxic. अगर this transition zone से ज़्यादा हो रहा है nutrient concentration in the plant system, then that is called sufficient zone. अगर sufficient zone भी से भी ज़्यादा हो रहा है, then that is excessive or toxic for the plant growth. Okay, so these are 4 different zones when we take plant nutrient concentration and plant yield or growth in a graphical manner. Deficient, critical, sufficient and excess. So in sufficient zone, another word is there, that is called luxury consumption. So plant is taking extra nutrient or plant is accumulating extra nutrient from the optimum, then that is called luxury consumption. It is mostly seen in maize and for potassium nutrient, because potassium is generally not combined with other organic molecules, so that there will be toxicity symptom. So their accumulation is higher or highest in the plant system, not highest in comparison to carbon, hydrogen, oxygen, but comparatively higher than other system, because there is no toxic symptom or no deficiency symptom if potassium is deficient or toxic, because they are not a part of different biomolecules. Steinberg effect, hidden hunger, and luxury consumption, they are very important. I have already discussed what is that. Steinberg effect means at the initial stage there will be nutrient dilution, low nutrient concentration will be there. Hidden hunger means nutrient is deficient but plant is not showing the deficiency symptom. Luxury consumption means without any toxic symptom, plant is taking up more nutrient than the optimum. Okay, let's discuss about deficiency symptom. What are the deficiency symptom you can find if particular nutrient is deficient? So in old leaves, if the nutrient deficiency is there in old leaves, there will be deficiency symptom of NPK, magnesium and molybdenum. Why in old leaf? Because when we see a plant system, we have already discussed about different nutrients and their based on their mobility in the plant. Okay, if any nutrient that is highly mobile, so after absorption and it will directly translocated to the younger leaves. Younger leaves means top leaves. Okay, as these nutrient NPK, magnesium, molybdenum, they are highly mobile, so they will translocated into the younger part or the newly developed part. So older part will get the deficiency symptom if there is certain deficiency happens. Then from NPK, magnesium, molybdenum, Dead spot you can find in if deficiency of potassium and molybdenum is happening, then there will be dead spot. Without dead spot, NP magnesium. Okay, and the green vein will be there. Again, from the NPK and magnesium, there will be intervenal chlorosis of older leaf you can find in magnesium. That means vein will remain green and the intervein area that will be chlorotic. In yellow vein or complete chlorosis will happen in nitrogen. Another one is, this is old leaves, then new leaves. In new leaves you can find deficiency symptom if there is deficiency of sulfur, iron, manganese and copper. Again, there is green vein you can find if the deficiency of iron and manganese is there. There will be intervenal deficiency or intervenal chlorosis in younger leaf. Then complete chlorosis if there is deficiency of sulfur and copper. Then old and new leaf both, the deficiency if there is deficiency of zinc, then that is you can find in old as well as new leaves. Then terminal boards, you can find the deficiency of calcium and boron in terminal board because they are immobile in plant system. If they are immobile, then that cannot be translocated into the top layer or board region. Then that is why the deficiency symptom you can observe in terminal board for calcium and boron. Okay, so this is an overall idea if any particular nutrient is deficient, where the deficiency symptom will be.